How old do you usually die if you have the Huntington's disease?
Middle age, like in your 40s. The problem with Huntington's disease is most people have had kids by the time they figure out they have Huntington's disease. And it's a dominant disorder. So, if you have Huntington's disease and you are... Hold on. Hold on. If you have Huntington's disease and you're big H, little h, and you marry a normal person, what's the odds your kids are going to have Huntington's disease?
1 out of 4?
1/2.
Oh wait, no, yeah.
You want to do Punnett Square? So, here's how I would do Punnett Square. Big H, little h, and little h. Why wouldn't I put 2 little h's?
Because you're wasting time.
Because it's a waste of time. So the kids are either have Huntington's or don't, 50-50 chance. That's often what you see.
Wait, how do you find out that you have it?
It's a nervous system disorder. You start losing control of your muscles in middle age, and people looks like they have Parkinson's. They shake, and when they walk, they have trouble, and it kills them pretty quickly. Yeah, well over 4 or 5 years, they die, and the kid sees them like that, and they know it's a dominant disorder, so the kid's got a 50-50 chance of developing that.
Do they, can you like painful test?
I don't know if it's painful. That's a good question. Yeah, there's a lady who developed a test for it. And she figured out a genetic test for doing it. And guess what? Her mother had it. So she sought out to get a test to figure out how to test for it to see if she had it.
Did she have it?
And she didn't. And her sister did.
Oh.
Isn't that crazy? The dominant lethal disorders are bad. Huntington's is probably the main one you hear about in humans. Okay. So, big Punnett squares. You ready? You're gonna need some paper here, something to write out some big Punnett squares on.
Dave.
I want a big Punnett.
You like a big Punnett square, huh?
Yeah. I'm just using it.
So Mendel got a lot more involved in his crosses than we've studied so far. If you take, okay, so big Y equals yellow seeds, little y equals green seeds.
Oh yeah.
Big R equals round seeds. And that's dominant over little r, which equals wrinkled seeds, which are my favorite pea seeds. I don't know. I just always, when I ate peas when I was a kid, they looked like that. And I like them.
I like peas too, as well as.
You know, I hadn't eaten peas much since I was a kid. Why do they give peas to kids and not to adults? They.
Children deserve to eat.
So here we go. We made a yellow round pea with a green wrinkled one. You're not mating the peas together. You're mating the plants that produce those peas. Right? Peas can't make, plants can make. So Mendel took the paintbrush and he moved pollen from one plant to another. A plant that produces yellow round peas mated with a plant that produced green wrinkled peas. And what do you think he got in the F1 generation? He got all yellow round, because yellow is dominant and round is dominant. But then he mated 2 of these with one another. And we call that a dihybrid cross. Why is it called di? Because there's two traits. Why is it called hybrid? Because you're crossing a dominant with both traits with a recessive for both traits. Dihybrid cross. Okay? So, you're mating big Y, little big R, little r, with another big Y, big R, little r. How do you determine the kids? Whenever you're doing a problem like this, you have to first determine the gametes. The gametes are the sperms and eggs. So I'm going to draw 4 sperm cells up here. We'll draw them with little tails to show that they're sperm cells. Even though plant sperm cells don't have tails.
Why not?
Because they're transported by the wind in a pollen grain. Or a bee in a pollen grain. They don't have to swim through fluid. Alright, so here's 4 sperm cells made by this parent plant. These are going to be the 4 pollen grains, let's say. So, remember, during meiosis, these genes split up and moved to opposite sides. Do you remember that when we had the chromosomes? They'd line up in the middle and then they could go to opposite sides. So the big Y and the little y alleles can split up. So a sperm cell is going to either get the big Y or the little y. It's not going to get both. It gets one of each pair of genes. So we'll say that it gets, this sperm gets the big Y. And the same with the R's. They split up in meiosis. So maybe it gets the big R. That's one possible sperm. Is there another one you can think of?
Big Y, little R.
Oh, big Y, little r could be in the sperm. What else?
Little Y, big R.
Little y, big R.
Little Y, little R.
Are there any other possible sperm cells? Why not a big Y, little y sperm cell?
Because you have like one of each letter.
The genes split up in meiosis. Each pair splits up. That's when the homologous pairs separate in meiosis. The big Y and the little y split. That's why you can't get a big Y and a little y in the same sperm cell. The big R and little r also split in meiosis. Okay? So those are the 4 sperm cells. Now this one would make egg cells. And we'll put the egg cells on this side of the square. What 4 egg cells could those make?
The same one.
The same as the sperm cells. But we could have 4 egg cells here with the same types of genes. Big Y, big R, big Y, little r, little y, big R, little y, little r. And so, how do we put these together? It makes this giant Punnett square that's also on the board. But I'm just going to show you a few squares so you can know how to do this. It's on the screen, I mean. So how do you put them together in these boxes? You take the 2 Y's and you put them together. So in this box would go big Y, big Y. And by the way, when you're doing problems like this, you need to make sure you can tell your big Y from your little y. Because if they look the same, which some of y'all have bad handwriting, if they look the same, you're going to screw these problems up.
I just have a bad hand.
And the 2 R's go together. Big Y, big Y, big R, big R. So what kind of pea plant will what kind of peas will it make?
Yellow and round.
Yellow round, just like the parents. Do me a favor, write down on your sheet of paper what would be in 1, 2, 3, 4. What would be in those 4 boxes? See if you can write them down. Actually, no. Let's not do this. Let's do these 4 boxes.
Grr.
There we go. You can just jot it down. You can just look at my thing, write 4 things. See if you can do it without cheating.
What's fun with that?
Figure things out in your brain.
Stop cheating off me, Mr. Willis.
Oh, wait a second. I got a hand back. What is this from yesterday? Come and get these when I call your name. Andrew. Chandler. Cartier. Dylan. Loretta. Mad. Nomad? She's a nomad. Jossie. Julie. Tammy. Meryl. Lottie. Lottie. Thomas. Charlie. David. Josie. No, David? Oh, there's David. All right, you get it? What goes in this box? Yellow, wrinkled. What goes in this box? Big Y, little y.
Heterozygous.
Big R, little r. Always put the dominant first. Don't write little r, big R. Write big R, little r. What goes in this box?
Heterozygous.
Look at you using scientific terminology. That's also a yellow round. What goes in this box?
Little y, and big R, big R.
That's a green round.
Yeah.
Y'all see how to do these?
Yes, sir.
So, when all is said and done, it'll look like this. And when you count up the kids, like, okay, so that, when you count up the individuals in the F1 generation, you get 1, 2, 3, 4, 5, 6, 7, 8, 9. You get 9 yellow rounds. You get 3 green rounds, 1, 2, 3. You get 3 yellow wrinkles, 1, 2, 3. And you get 1 green wrinkle. We call that a 9 to 3 to 3 to 1 ratio. And that's what you get when you have a dihybrid cross. That's what you get in the F2 generation. It's always a 9 to 3 to 3 to 1 ratio. If you just memorize that, you won't have to do this whole Punnett Square. Memorize that. It's going to be in a 9 to 3 to 3 to 1 ratio. If you do a dihybrid cross, which is they're both heterozygous for both traits. Now, let me give you some problems.
No, oh my God, no problems.
Where it's not exactly like this. I mean, what if you had big Y, little y, big R, crossed with little y, big R, little r? How would you do this problem? This is the kind of thing they'll give you on the test. First of all, what does that one look like?
Yellow and round.
It has a yellow round phenotype. What's the phenotype of this one?
Green.
Green and round. So what are the kids going to look like?
Yellow round.
Well, first thing we got to do, like we did before, is we got to come up with the gametes. We got to come up with the sperm cells that this one can make. Give me one type of sperm cell it can make.
Big Y.
Big Y, big R.
Big Y, big R.
Yes, sir.
What else can it make?
Why not another big Y, big R?
We don't have to do another one because we already got one.
Fascinating.
We just do all the different types of gametes that it can make.
Little Y, big R.
What?
Little Y, big R is another one. Is there anything else it can make?
Little Y, big R.
Those are the only two possibilities. So, what about this parent? We'll put this parent on the side of our square. So here we're making a square. This parent's gametes go on the side. What could it make?
Little Y, big R.
Yep. And what else?
Little Y, little R.
That's all it can make.
Yay.
Now let's do that Punnett Square. What goes in this box?
Big Y, little Y. Big R, big R.
And what is that?
Yellow.
Yellow round. Little y, big R, big R. Y'all see how I'm doing this? Little y, big R, big R. What is that?
Green round.
Green round. Big Y, big R, little r. What's that?
Yellow round.
Yellow round. And finally, little y, big R, little r. What's that?
Green round.
So half, it's going to be a 1-to-1 ratio. It's going to be one yellow round to one green round. There, we've done that problem. Now, you need to learn some shortcuts.
Do we?
Yes.
Because I can do this in my head immediately upon saying it. I'm that good. And you can be too.
Well,
Teach me,
Learn
Master.
from my wise ways.
Yes,
Teach me, Sensei.
Here's how you do it. Do you see right away, when I look at this, I see right away all the kids are going to be round. How do I see that?
there's no way for it to be homozygous
See the
recessive.
two big R's there? Doesn't even matter what these are. These could be big R, little r, little r, little r. If I see two big R's here, we know all the kids have to be round because R is dominant. Round. All the kids are going to get a big R from this parent. All the kids are going to be round. I know that immediately. I can forget about the R's and just do the Y's. I could just do big Y, little y, mated with little y. And my possibilities are big Y, little y, little y, little y. Half yellow, half green. So half my kids are going to be yellow round, half are going to be green round. They're all going to be round. So as soon as I wrote that, I knew the answer in my head. Because I know a big Y, little y crossed with a little y is 50%. You might have to do that out. I just know it from doing a lot of these problems. So what if I changed it like this? What if I wrote this and it was that? Can you get it in your head? Or maybe do it on a sheet of paper.
3 yellow and 1 green.
Big Y, little y crossed with big Y, little y is a 3-to-1 ratio. That's a 3-to-1 ratio. So we end up with 3 yellow, there they are, to 1 green. So since we got the big R here, they're all going to be round. So it's 70, it'll be a 3-to-1 ratio, yellow round to green round. That's what this will be. If we do that. Want me to give you something to try?
Yeah.
What do you mean no? All right, try this one for me. Mate, little y, big R, little y, with big Y, little r, little y. Do that on your sheet of paper. Probably have to use paper unless you're good. No, you can't do it like that because you have to tell. Yeah, you have to put them together. That is technically correct, but you have to tell the percentage of each. You're on your way. You're on your way. You're not starting?
No, I started. I just restarted.
But is that the right answer? If that's the right answer, yes. Tell me what the phenotypes are. So what color are they? What shape are they? You're on your way. You are not on your way. You copied it.
Oh, it did? Oh, it is too.
Huh?
That would be it.
That's one thing, you gotta copy it right.
Can you skip out on that?
Yes.
Okay.
Chelsea, you're on your way. Meryl, you're on your way. I think you copied it wrong. Oh, it's here? Oh, you are on your way. It's kind of messy and hard to read, but it looks like it's on the way. Tell me the percentages of the kids, the phenotype.
Now?
Now?
No, I mean write it down.
Oh. You want to check?
Sperm of this plant. Little y, big R, little y, little r. Did y'all get that far?
Yes.
Eggs of this plant. Big Y, little r, little y, little r. It's going to be a 4x4 or 2x2. So, big Y, big R, little r. What's that look like?
Yellow round.
Yellow round. Big Y, little r, little r. What's that?
Yellow.
Yellow wrinkle. Little y, big R, little r. What's that? Green round. Little y, little r.
Green wrinkled.
Green wrinkle.
Ooh, nasty,
That's a 1 to 1 to 1.
1 to 1.
It's a 1 to 1 to 1 to 1 ratio.
Wow.
I got another one for you.
Grr.
This one, I want to see if you can do it fast. Pay attention.
It's okay.
Oh.
You should have it done already.
Got him. Yeah. Oh, well, now we do.
Did you get it done already?
Yep.
Didn't have to write a thing when you see that. The two big yellows over here, that means all the kids are going to be yellow. Doesn't matter what's over here. The two big R's over here, that means all the kids are going to be round. 100% yellow round.
Wow.
Isn't that fast? Most kids would spend 3 or 4 minutes on this one, doing Punnett Squares. I'm serious. This is why they don't finish the test. You got to be able to kind of figure these things out. We're doing a problem sheet on Friday. I'm going to give you a problem sheet with a bunch of these and we'll work on them, okay? There might be some on a possible quiz. What about a tri-hybrid cross?
That sounds like a lot of work.
I just won't be doing that.
What if we added,
I didn't know you could do that.
check this out, big Y, big R, little r, and big T, little t. It's yellow, it's round, and it's tall. And what if we made it with another yellow, round, tall? What kind of Punnett Square is that going to look like?
It's not the one that I want to do. Really?
It won't have. So when there was one trait, the biggest Punnett Square we could get was a 4-box. When there was 2 traits, the biggest Punnett Square we could get was 16 boxes.
30.
So how big would the Punnett Square be when there's 3 traits?
64.
One trait is 4 boxes. 2 traits is 16 boxes. What would 3 traits give you? We got any mathers in here? You know, did I mention I was on the state championship math team? I may have never said it.
Yeah, 64.
36.
It's 64.
Oh my God, I was right.
Good job.
Get
You
out.
put 4 to the power.
Too much work. I'm not doing that. Answer the door, then.
4 to the first, 4 to the second, 4 to the third.
I just won't be drawing that. So,
4 first, 4 to the second, 4 to the third. So, are you going to draw a 64 square Punnett Square and sit there and work it out on the test?
No.
It'll take freaking forever. So you can do what's called the forked line method. You can figure out, for instance, for yellow, that it's going to be 3 yellow, 1 green. See how they do there?
No.
They put at the top that if you're just considering yellow, there's 3 yellow to 1 green. And then on each side, you have to consider round. There's 3 round to 1 wrinkled. If you do big R, little r cross with big R, little r, you end up with a 3 to 1 ratio, right?
Yeah.
So you put 3 round to 1 wrinkled over here, and you put 3 round to 1 wrinkled over here. And the same thing with tall and dwarf. Tall and dwarf is a 3 to 1 ratio. So you put, for each round, you put 3 tall, 1 dwarf. For each wrinkled, 3 tall, 1 dwarf. The round here, 3 tall, 1 dwarf. The wrinkle here, 3 tall, 1 dwarf. This is called the forked line method. And so, you just multiply them together. What's 3 times 3 times 3? There's going to be 27 yellow, round, tall. There's going to be 9, that's 3 times 3 times 1, yellow, round, dwarf. See how this works? So, you can find out that it's a 27 to 9 to 9 to 3 to 9 to 3 to 3 to 1 ratio.
Got it.
How would this change if I did this? What would I change about the whole thing if I did this? If I made that line there.
Oof.
Would it make it 2 to 2? Well, it makes
What would we change about this whole forked line method?
the yellow to green part would say 2 to 2.
This would say 2 to 2. But can you make it? What's 2 to 2 the same as?
1, 1.
1 to 1. So it's the same as the 1 to 1. So I could change this yellow from 3 yellow, 1 green. I could change it to 1 yellow, 1 green, and then I could easily get my results from changing that Y. That's the forked line method. You don't have to use the forked line method. There's other ways to do it. This is a 2-day section 12.3. So I'm going to leave it right there. Why don't you try to read through when they do the statistical parts here. We'll just leave it there. Do what?
I said Willis out.
Willis out.